Hey, happy Monday. Welcome back from the weekend. I hope you had a good one. I did. If you're watching this, you must have already done that bell ringer. Notice we're doing a little bit more now. Don't just put the answer. You're going to give your answer and tell me why you know that answer is right. Because why do the least when you can do the most, right? So now, if you have done that, that's why you're here. We're going to dive into direct instruction. So make sure that those notes are pulled up and you're typing as I type. So what are we doing today? Well, on Thursday and Friday, we talked about mitosis. Not too detailed, but we just kind of eased our way into it. So this first slide, look at that, I've already got it filled out for you. You're welcome. Just a reminder of what we talked about. So mitosis is a way that cells repair damaged cells, they make new cells, they help you grow. We're just making new body cells. So there are 4 phases of mitosis: prophase, metaphase, anaphase, telophase, or we remember PMAT, P-M-A-T. Today, that's what we're going to talk about. We're going to go into more detail about what each of those phases, like what are they? What is the deal with mitosis? What does it even do? So mitosis helps us, like I said, make identical copies of our cells or the genetic material in the cells. So all of your genes and chromosomes inside your cell is being copied identically, and it passes that down to the new cell. Anytime something is being reproduced identically, it's an identical copy, that's known as asexual reproduction. And remember, this occurs only in our body cells, the somatic cells, not our sex cells. We'll talk about those a little bit later. Okay, so before we get too into the details, let me move myself. Let's go down here. That works. Before we get too into the details of what's going to happen during mitosis, we have to cover this super important part. It can get a little complicated, but I think once we break it down, it's going to make sense. So, before cell division even happens, your DNA has to be copied. So, copied, and it's packed into the X-shaped chromosomes that we've talked so much about. Okay, so before they are copied, this right here is what it's going to look like. When it's just, it kind of looks like a stick or number 1. When that is what it looks like, that structure is called a chromatid. Oh my gosh, chromatid. It's Monday morning, you guys. Okay, so that chromatid is then copied or duplicated, same thing. And that is what is going to make that X shape. It's going to be that first chromatid and its new buddy, its identical copy chromatid. And they are called sister chromatids because they're little sisters, they're twins. They look just alike. So once they are hooked together, that's going to give it that X shape that we've talked about. And those copies are going to be held together by that little, it looks like a little button in the center that is called the centromere. That's easy to remember that that's its name because it sounds like center. It's right in the center. Okay, so one chromatid duplicated, then we have our sister chromatids, and that makes the chromosome. That happens before mitosis. All right, so let's actually get into the phases that we've talked so much about. Phase 1, that is our prophase. So what happens in prophase? So the chromatids, remember that's those little stick guys, the chromatids in the nucleus are going to condense. Ooh, I'm in all caps. They're going to condense or compress, kind of get close together, shrink. That makes them nice and thick. So once they thicken up, they're actually big enough that you can see it underneath a microscope if you have one. Also, what happens is that membrane around the nucleus breaks down. So you can kind of see that happening right here in this little GIF. So that's the nucleus, is that pink circle. It's actually going to break down, so they're no longer kept right inside there. They're free. They've been released. So you can see an interphase. Remember, that's when your cell's just doing its normal job. It's not going through mitosis. It's doing what it's made to do. This is how it's going to look. And then after that DNA is copied or duplicated, it goes into prophase. And you can actually see your chromosomes nice and thick right here in that X shape. Down here, we are going to have a word to help us remember what happens in each phase, because it can get kind of tricky. So when you see the P in prophase, we're going to remember prepare, because this is step 1. Everything's breaking down. It's getting all ready to go through mitosis. So when you see P, think prepare. I know what's happening in prophase. Moving on. This is my favorite phase because I think it's the easiest. So phase number 2 is called metaphase. Metaphase. So what happens in metaphase? The chromosomes, those little X-shaped guys, they are just going to line up right in the middle. I cannot spell. In the middle of the cell. So if you see in this little picture here, they go, wait for it. They move and move and move and move and bam, make a beautiful single file line. Okay, so when that happens, the centromeres, or remember the little tiny part that's in the middle of the chromosome holding those chromatids together, they are going to be the same distance from each side of the cell. So in other words, the chromosomes are exactly in the middle. Okay, so how do we remember metaphase? Easy. We see the M and we think M for middle. They're right in the middle. See, the easiest one, I swear. Okay, next one, moving myself down again, is anaphase. Anaphase. Okay, so what happens in anaphase? This is where things get a little wild. So the sister chromatids are actually going to separate. Kind of sad. I know, they're separated. So once they separate, they are pulled away to opposite sides of the cell. Tragic. So you can see right here, they are no longer together. They are moving away, all the way to the edge of the circle, to the edge of the cell. So then each side of the cell is going to end up with a group of chromosomes. So over here we're going to have one group, and over here we're going to have one group. When we see the A, we think away. They are just moving away from each other to the opposite ends. One thing I want to hit on real quick before we move on, I think this is like the trickiest part of mitosis to fully grasp. So we remember when a cell is in interphase, it has a certain amount of chromosomes, 23 pairs of chromosomes to be exact, and then they're duplicated. So after they're duplicated, your cell actually has more chromosomes present than normal because we doubled the amount, right? So now, once we have separated those chromosomes, each group now has the same amount as a normal cell. So one group has 46 chromosomes total, the other group also has 46 total. I hope that makes sense. I think it's the trickiest part of all of this to get. Last phase. Starts with a T, that is telophase. So during telophase, we're really wrapping it up. A new nuclear... I'm so sorry that you guys have to watch me just try to spell all day. That's oof. A new nuclear membrane is going to form around each group of chromosomes. That's just a fancy way of saying a new nucleus is going to form, because once we have those two new cells, they need a nucleus. Those chromosomes can't just roam free. So during telophase, that's when we get that new nucleus. So the cell now has two nuclei. It's just a fancy way of saying nucleus, plural, nuclei. Then the chromosomes become less condensed. So they are not quite as thick. They kind of stretch out a little bit more. You can't really see them under a microscope anymore. Way to remember telophase, T. T stands for two. Two nuclei, nucleuses. Okay, so you might be thinking, Miss Long, that's all the phases of mitosis, and we still don't even have two cells. We're still in one cell. Something happens after mitosis. We hit on it a little bit last week. After mitosis, there's that big fancy word again, cytokinesis. Cytokinesis is going to happen. It's when, just like in this GIF, you have your two cells forming because they split off from each other. They just can't take it anymore. They gotta separate. So you're going to get two new identical cells, and they're identical because their genes are exact copies of each other. All right, let's go back through it one more time, just a quick wrap up so we remember what's going on. Remember, before we do mitosis, your DNA is copied, so you have that one chromatid, it becomes copied, and you get the X-shaped chromosome, or two sister chromatids together. In prophase, we see P, prepare. We're preparing to go through mitosis because that nucleus breaks down and those chromosomes condense and get nice and thick. Metaphase, M for middle. Those chromosomes just line up nice and straight in the middle, same distance from opposite ends. Until we hit anaphase, we see the A, we think away. The sister chromatids separate and they move away from each other to opposite ends of the cell. And then we have telophase, where that new nuclear membrane forms around those new groups of chromosomes. So our two nuclei are going to form, and we see that T, we think two, for two nuclei, whatever. Then it becomes two cells because of cytokinesis. It's when they finally split off and you have two cells with identical genes. Whew. All right, mitosis can be a lot. Honestly, between you and I think it's the hardest thing we learn all year, but that's just me. So if you have any questions, hit me up. That's why I'm here. I can answer them for you. I can walk you through it. We'll get this together. Tomorrow we're going to practice it. So for now, you're pretty much done. Have a good one.